Now let's take a closer look at what actually happens during the process of photosynthesis, so how we're actually going to make sugar. So photosynthesis is an anabolic endergonic process. If you'll remember that anabolic means we're adding things together to build more complex molecules. Endergonic means that it's a process that requires an input of energy. So in photosynthesis we take carbon dioxide and water, we add hydrogens that were originally on that water to the carbon dioxide, making our glucose molecule. Okay? Remember, when you're adding electrons, when you're gaining electrons, that's reduction. Remember oil rig? So reduction is gaining of electrons. And then our oxygen in the water molecule is losing these hydrogens, so it is being oxidized. Photosynthesis can happen in chloroplasts, or it can happen in photosynthetic cell membranes. So for prokaryotes that do not have chloroplasts, they can still do photosynthesis if they have photosynthetic regions of their cell membrane. So just a quick review of chloroplast structure. A chloroplast has two membranes. It has an inner membrane or outer membrane on the outside, and then an inner membrane on the inside. The space between the two membranes is called the intermembrane space. So the white space between the two orange lines is the intermembrane space. Now, inside of the inner membrane is a gooey liquid called the stroma. Okay? The stroma contains these stacks called a granum. So each stack is called a granum, but each individual thing that is stacked, so each individual pancake or coin, you know, they look like stacks of coins or pancakes. So each individual coin is called a thylakoid. Now, photosynthesis is a two-part process. It first requires an input of light, and those are called the light reactions. And then the second part does not require light. It doesn't happen necessarily in the dark. We don't call it the dark reactions. We call it the Calvin cycle for the scientists that discovered it, or discovered how it worked. So our light reactions occur in the thylakoid membranes. They actually occur in these little stacks. Okay, light is used to drive the production of ATP and NADPH, which is reducing power. So the whole goal of light reactions are to produce ATP and NADPH. So the ATP is going to drive the Calvin cycle. And NADPH is just an electron shuttle. So it's going to carry electrons to the Calvin cycle. Water provides the electrons that are needed and is converted to oxygen gas, a waste product. So water is required for this process, and it releases oxygen gas. Now, the Calvin cycle occurs in the stroma, so it occurs in the liquid outside of the thylakoids. And the ATP and NADPH, which are produced during the light reactions, are used to incorporate carbon dioxide. So it's taking carbon dioxide and incorporating it into a 3-carbon sugar. Now, it's not making glucose right away. We'll discuss that in a little bit, but it's actually making a 3-carbon sugar called G3P. Alright, so let's discuss the light reactions. Our next video will discuss the Calvin cycle. So light plus chlorophyll causes a release of electrons. So when photons of light, so basically think of it like a molecule of light. Light is a wave, but we can also think of it as matter too, and we call them matter of light photons. So when a photon of light interacts with chlorophyll, okay, one of its electrons becomes excited. So when chlorophyll interacts with the light, one of its electrons becomes very, very excited, and this happens with about 1% of all light that reaches Earth's surface. So now we need to take a look at photosystems, because they're very important for the light reactions. They're where they actually occur. So photosystems are complexes of protein and pigment molecules. So they're protein with pigment molecules inside of them or incorporated into them. And they are embedded in the thylakoid membrane. So remember the little stacks? They are protein and pigment embedded into the stacks. Okay, photosystems direct incoming photons of light to the reaction center, where chlorophyll A molecules produce excited electrons. And those excited electrons are then transported to an electron transport chain. So the photosystems, they take photons of light and they transfer them from chlorophyll to chlorophyll, or from pigment to pigment to pigment, to 2 special chlorophyll molecules. And those special chlorophyll A molecules have an electron that's been excited. So if the photon is transported along or through these different pigments, And finally, they're directed to these chlorophyll A molecules. And those chlorophyll A molecules have their electron excited, and then they pass them on to something called the primary electron acceptor. Okay, there's two types of photosystems. We have photosystem 2, and that works at the start of the electron transport chain. It's at the very beginning of photosynthesis. And then we have photosystem 1, which happens at the end of the electron transport chain, so at the end of the light reactions. Now, the reason the numbers are different is because photosystem 2 was discovered after photosystem 1, but photosystem 2 actually occurs sequentially before photosystem 1. All right, so let's take a closer look at our photosystem. So this is our thylakoid membrane, the phospholipid bilayer that makes up those stacks inside of a chloroplast. So inside of the phospholipid membrane, or the phospholipid bilayer, we have proteins. Okay, that's the red structures. Now those proteins contain pigment molecules, chlorophyll, and other pigments. And when a photon of light reaches those pigments, they pass that photon, that energy, from pigment to pigment to pigment. Eventually, it reaches special chlorophyll A molecules that are located in a region called the reaction center. So this orange region is called the reaction center. And then those chlorophyll A molecules pass their electron to a primary electron acceptor. Okay? And so this is like an overview of what our photosystems do, and then we'll talk about why this is important in our next slide. Alright, so let's talk about how electrons actually flow in our photosystems. So let's start with non-cyclic electron flow. So in non-cyclic electron flow, electrons move from photosystem 2 to photosystem 1 via the electron transport chain. So photosynthesis uses the electron transport chains just like cell respiration does. So from photosystem 2, electrons are transferred to the enzyme NADP reductase, okay? And then that uses those electrons to reduce NADP into NADPH, okay? Or NADPH. And non-cyclic electron flow produces both ATP and NADPH, okay? And it also requires an input of water. So this is our photosystem 2. Again, we're taking light, and a photon of light is transported from pigment to pigment to pigment, and is eventually transported into two special chlorophyll A molecules, which pass their electron to the primary electron acceptor. Now, they have lost an electron, okay, and so they need another one. They get their electron from water. So water passes 2 electrons to these 2 chlorophyll A molecules to fill their electron void, okay, and then the oxygens are released as oxygen gas. Now, that primary electron acceptor passes its electron to something called plastoquinone, or PQ, okay, and then that electron flows through an electron transport chain. These are increasingly electronegative molecules. So it passes an electron from plastoquinone, or PQ, to a cytochrome complex, to something called plastocyanin. If you want a review of what the PQ and PC are, it's right over here. Okay? So our electron is flowing down to increasingly electronegative molecules, and that causes a release of energy, or ATP, or we use it to synthesize ATP. And then the electron from plastocyanin is eventually going to move to these chlorophyll A molecules here. So let's take a break from that electron transport chain and talk about another input of light in photosystem 1. So photosystem 1 is going to absorb light, and a photon of light is going to be transported from pigment to pigment to pigment, and eventually moves to the reaction center and the chlorophyll A's in that reaction center. Okay, they are going to take their electrons that were excited because of this light, and they're going to pass it to their primary electron acceptor. Now, they also have an electron void now. We don't have an input of water here. They get their extra electron from the electrons that are passing through the electron transport chain. So these electrons stay here, and the electrons that were excited due to the light move up to the primary electron acceptor. Now, the primary electron acceptor passes its electron to ferredoxin, or FD, and the ferredoxin then passes an electron, or 2 electrons, to NADP reductase. Okay, and NADP reductase is going to take NADP and convert it to NADPH. All right. So again, what we're producing here is ATP, and we're making NADPH. All right. So sometimes our electrons flow cyclically. We call that cyclic flow, and this It involves an electron flow from photosystem 1 to the electron transport chain back to return to photosystem 1 again. Okay? It only produces ATP. It does not require an input of water, and the whole reason we need to do it is because the Calvin cycle requires more ATP than it does NADPH. All right, so it's requiring 9 ATP with 6 NADPH to make glucose. So we need to get an extra supply of ATP. All right, so we're going to have our electrons that are excited due to light. Okay, and those electrons are passed to our chlorophyll A. They pass their electron to the primary electron acceptor, which normally goes to ferredoxin. I drew ferredoxin on this side on our previous slide, but it can be located anywhere in this complex in photosystem 1. Now, instead of ferredoxin transporting its electron to NADP reductase and making NADPH, in cyclic electron flow, instead of transporting its electron there, it transports it back to the cytochrome complex, which is part of the electron transport chain, which then passes it to our plastocyanin. This is supposed to be PC here. Now this is an electron transport chain, so it releases ATP, but we didn't make any NADPH in this process. So cyclic electron flow's goal is to make more ATP. Now let's talk about how ATP is actually produced due to the light reaction. So we're going to use chemiosmosis to make ATP just like we did in cell respiration. So this represents our thylakoid. The thylakoid are the stacks inside of a chloroplast. Now inside of the thylakoid is called the thylakoid space, and outside of the thylakoid is called the stroma. All right, so as electrons move through the electron transport chain, they provide the energy, or a proton concentration, for chemiosmosis in a fashion that's almost identical to cell respiration. So we're taking light, okay, it's passing through our photosystems just like we discussed in previous slides. Now, that light energy is used to pump our electrons, or pump hydrogens, or protons, into our thylakoid space. And then those protons are going to flow through ATP synthase. Remember, ATP synthase is also involved in cell respiration. Now, we're going to have a high concentration of protons inside the thylakoid, and they're going to rush through the ATP synthase to the low concentration of hydrogens that's in the stroma. Now, that rush of our protons is going to power the production of ATP. Okay, so we're going to add ADP and phosphate and use that flow, that energy of the flow of electrons or protons through our ATP synthase to make ATP. Now, there's a couple of differences between cell respiration, or chemiosmosis in cell respiration, and in photosynthesis. In respiration, the energy comes from the oxidation of glucose. So we're oxidizing glucose, and that's where we get our energy to power chemiosmosis. And we call that oxidative phosphorylation. But in photosynthesis, the energy comes from photons of light, and so we call it photophosphorylation. We're using energy from light to phosphorylate ADP and turn it into ATP. Also, in cell respiration, protons were pumped into the matrix, kind of like in the gooey part, the outside part of the mitochondrion, by the electron transport chain. But in photosynthesis, the protons are pumped from the stroma into the thylakoid space, and then they rush back out to the stroma, and that rush produces ATP, or is used to power the production of ATP through chemiosmosis. Alright, we're almost done now for an overview of what we produce in the light reaction. So a quick recap. Our inputs were light, so we get our photons of light. We have ADP and inorganic phosphate, NADP, and water. Okay, remember our water is our source of our electrons. And then our outputs were ATP, so energy, and NADPH, and then oxygen gas. The ATP and NADPH are going to go to the Calvin cycle, and the oxygen gas is released as a waste product.